So trypanosomes are masters of disguise. Indeed, they undergo antigenic variation in their mammalian hosts in order to evade the host immune response. And antigenic variation is a highly sophisticated and very fascinating virulence mechanism that is actually employed by a great number of pathogens that include multiple bacteria, as well as parasitic protozoa such as trypanosomes or malaria-causing parasites. And antigenic variation, as you can imagine, greatly challenges vaccine development against these organisms. So these pathogens have these large families of genes that encode for surface-exposed antigens that they can shift through genetic mechanisms. But a key component for successful antigenic variation is the ability to express a single antigen at a time, which we can refer to as monogenic expression or single gene choice. And this is incredibly important so that they don't prematurely exhaust their antigen repertoire. But single gene choice is not something exclusive of organisms that undergo antigenic variation. In fact, many genes in our body are expressed in a monogenic fashion, and this is not fully understood in any eukaryote. In the case of trypanosomes, they are quite extreme in their biology. So they have this variant surface glycoprotein, or VSG, and they express a single one from thousands of possible genes. So it's the largest known gene family in nature. And the single active VSG makes the most abundant protein in the cell because it completely covers their entire cell surface. And they are extracellular, so they are fully exposed to the host immune system, and they are in a constant battle against adaptive immunity, which they often win. So they can infect humans and other mammals for years, even decades, with continued waves of parasitemia. And the machinery responsible for this VSG singular expression have remained elusive for decades. But recently, I have identified this novel protein complex that I call the VEX complex that regulates this process. So when I deplete these proteins in these parasites, they start simultaneously expressing multiple VSGs, and therefore they are rapidly eliminated by the immune system. In a more recent follow-up work, I've discovered that this complex engineers a very specific DNA arrangement in the nucleus, so it leads to the formation of a specific compartment where a single antigen coding gene can access RNA processing factors. So in simple terms, it's the assembly of a VSG transcription and splicing superfactory that on the one hand allows the expression of a single antigen while simultaneously making sure that that antigen is expressed at very high levels. So in the future, I'm really interested in understanding how these transcription factories are assembled and how proteins such as the VEX proteins operate. And hopefully that will elucidate mechanisms underpinning antigenic variation, which is crucial to sustain infections by trypanosomes and other deadly pathogens. And in a broader context, trypanosomes are very good model organisms to study single gene choice, which remains one of the biggest mysteries in eukaryotic gene expression. Thank you.